Okay, so another experiment that we teach our students in Biosciences lab is a technique called Polymerase Chain reaction. So what Polymerase Chain reaction is, it's a way of being able to amplify DNA. So for example, how does this work in real life? If there was a sample of DNA recovered at a crime scene, that DNA, that small amount of DNA can be amplified into lots of DNA, which would then be analysed to try and work out who the perpetrator was. So it can be used in crime scenes, but it can also be used for genetic studies as well. So this is something, a fundamental technique that we teach our students. So this is the piece of equipment that allows us to do this. This equipment is called a thermocycler. So what our students would do is they would set up their samples. Again, they can set them up in a 96-well plate, but we can also set them up individually. The plate would then, or the tubes would then be put into the machine, and the samples would then be run. So this particular process mimics how DNA is replicated within our cells in the body. Okay, so the students can kind of put into context what's happening in our cells in terms of that replication process to actually what's happening in the lab. Once that DNA is amplified, the students would then carry out some further analysis using a technique called gel electrophoresis, which would then allow them to see whether they have any DNA present or not.